Hi, good morning. I'd love to have gone live on this, but for some reason my internet's a little bit slow, so unfortunately I'm going to have to record it. Today it's 7:30 a.m. On the 16th of June, at least in London time, it's 7:30 a.m. I've had to put these slides together rather quickly because I have to catch a flight today to Norway. I'm sure some of you or many of you may know, and we have a significant presence in Norway, so I'm going to have to go there today. So my apologies for going early, having to record, and also having to go a little bit fast as well. So some of the questions are about wire sensors, so I'll answer the questions about wire sensors. Some of the questions are about making immunosensors. Somebody's trying to make an immunosensor in the UK. They're using streptavidin pre-coated electrode. I will discuss that. And somebody else is asking about customized screen printed electrodes, and I'm going to advise on that as well. With that said, let me go forward. If any links pop up today, by the way, I'll make sure I put them underneath the video as well. We do have some webinars coming up this week. So we've got one on point of care testing on the 11th of July. I say this week, this is actually a month's time. So if you're interested in wearable biosensors, please come to this webinar on the 11th of July. If you see a link, I'll put the link underneath the video and that will take you to the registration page. And I say wearables. No, this is actually on the 11th of July. We're doing a webinar actually on commercialization. So if you're interested in the commercialization, when I say commercialization, I'll just put my hand on it now. We have something we call Sense It All. It works with our screen electrodes and with our biosensors. It's got specific applications that are driven through QR codes. So glucose, chili, potassium, pH, for example. But we're also now extending it so that it actually is much more of a research instrument as well. So I've got a QR code. The significance of these QR codes is that the hardware is run through the smart device or smartphone, and the hardware then is told what method files to use by using QR codes. So we've got amperometry here, we've got square wave voltammetry here, we've got linear sweep voltammetry here, we've got cyclic voltammetry here, we've got open circuit potential here, and we also have electrochemical impedance spectroscopy. What I would suggest is the webinar, which I've got on the screen at the moment, is called Sense It All. That's on the 11th of July. That's a good one to come to understand this Sense It All platform. There's a wearables webinar next week. If you're interested in wearable biosensors, then please come to that. But I'm thinking a little bit about questioner number 2 and questioner number 3. Question number 2 is interested in immunosensors. Do come to this webinar on the 25th of June. And question number 3 is interested in custom screen printed electrodes. Do come to this webinar on the 25th of June. I'll make sure I put these links underneath the video when I've uploaded it. So question number 1 is about wire sensors. I like to obviously animate a lot of my answers, but with the earliness of the day and with the shortness of time I have available today, I've had to be a little bit more quick. Can the wire sensors operate at 3.3 volts rather than 0.6 volts? The quick answer is, if we're using an oxidase enzyme, lactate oxidase, glucose oxidase, then they give out hydrogen peroxide. It's the same if you're testing hydrogen peroxide directly. 650 millivolts is a good potential at which to oxidize hydrogen peroxide. If you use greater than that, you will end up doing the oxidation of hydrogen peroxide plus other molecules. Voltage, so rather potential in the units of voltage, is really energy. So the more energy you put into that system, the more molecules you can oxidize. So if you come away or go higher than 0.65 volts, then you will start co-oxidizing other molecules. Once you start getting towards 1 volt, you'll start doing hydrolysis of water. So 3.3 volts is disastrous. You'll get lots of current and you'll get lots of gas bubbles, and you're really doing hydrolysis of water. And the answer to your question is, you can't really run these biosensors at 3.3 volts. You'll put lots of current through them. Current and voltage is power. Voltage times current obviously is power. So you'll put too much power through them. It'll be non-specific because it will be just oxidizing everything. You'll end up doing hydrolysis of water, and you'll probably find that your electrodes go all blackened because actually they get oxidized by it. So that's my advice on question number 1. With the ATP sensor, yeah, you need glycerol in there in order to make the ATP sensors work. Is a quick answer on that. Question number 3. You can get a calcium biosensor. We've probably pulled it off the website. The guys are kind of, when I say the guys, they're trying to rationalize. We had thousands of products. They're trying to rationalize at the moment. So sometimes they're taking things down. So maybe they've taken it down and maybe we can put it back up again. So the calcium wire biosensor, we probably can do it. It probably says contact us, and that's probably what you're trying to do. So we probably can do it. It's just, we did get to a point where we had thousands of products and we're trying to be a bit more rational now. How many times can you use a wire sensing solution? Is it similar to the screen printed electrodes? Yeah, you could sort of rinse them off with DI water and reuse them. Is calcium amperometric or potentiometric? It's potentiometric. Lots of these ions fall into the category of, or we detect them using what we call ion selective electrodes. What is the role of increasing voltage for the biosensor or screen printed electrodes? The voltage, I feel like there's something out there. It's not very good because I'm a chemist, so I'm giving a chemist's analogy here. But voltage is a bit like wavelength. If you want to detect some molecules, you choose the right wavelength. I would say that voltage is a bit like wavelength in that respect. That if we want to if we want to detect a specific molecule, we choose the right voltage. So it's not one of these games where I just increase the voltage and I get more and more signal. It's really not like that. If you increase the voltage, you'll end up oxidizing everything or end up reducing everything, depending on which direction you go, as I've answered in question 1. So there is not an advantage necessarily to just keep on increasing the voltage, as discussed in question number 1. What is the detection range for nitric oxide and ORP, hydrogen peroxide, ATP, calcium? Let me have a quick look. As I say, this is not the way I would normally do the webinar, but I've literally just at the moment, just this morning, kind of running out of time. So let's have a quick look at this. Products, biosensors. Now, admittedly, I'm probably using screen printed electrode work here, and I'm sorry that is zoomed. So I'm just going to zoom out here. So the detection range for me looks like we're at 0 micromolar, 9 micromolar, 18 micromolar. So that gives you the sense of our detection range. That's the data that I have, whoops-a-daisy, in the public domain on nitric oxide. Let me have a look at your other question. ORP, oxidation reduction potential. ORP is not a molecule, it's a property of the solution. So oxidation reduction potential, it's weird, I know, but it's a property. It's the sum of all the antioxidants in solution. So if you start an experiment, you only measure the ORP, and then you turn off on the source of an oxidizing material, the signal should really go up, especially with what you're doing, which is quite high power. So ORP, oxidation reduction potential, is not the measurement of a particular analyte, it's the property of the solution, and it's the total number of oxidants or antioxidants in the solution. So you have to really get your head around what ORP is. It's not an analyte, it's a property of a solution. Hydrogen peroxide, that's the easy one to answer, because hydrogen peroxide is an analyte, so I can just then go and look up the analyte, hydrogen peroxide. I think hydrogen peroxide, we're probably pretty... we're definitely, this is something that we're definitely doing at ZP. We're testing things at 0.5 micromolar, 2 micromolar. So you can really get fairly low on hydrogen peroxide. I suspect it's quite sensitive. What's the next question? ATP, adenosine triphosphate. Let me have a quick look at ATP. Adenosine triphosphate. Where are you? ATP. There we are. Cool. There we go. Right. I'm going to have to zoom in a little bit in a minute. But I can see here micromolar. So let me have a quick zoom in. So here I can see that I can see 20s here. That's probably 10, 5, possibly, yeah, probably 5 here, 4, 3, 2, 1-ish here. So what can I say on this? We're probably detecting down in the 1 micromolar limit on ATP, adenosine triphosphate. Whoops-a-daisy, I've just went in the wrong direction. But calcium, right, calcium, whoops-a-daisy. I've just done something wrong and I need to fix it. Right, calcium, so let me just go to calcium quickly. By the way, the reason I use the screen-printed electrodes is because we use the same formulations for screen-printed electrodes and for wire electrodes, so they are representative. I've got a suspicion here, I'm looking at 0.2. By the way, we develop most of our sensors to work in the clinically useful range. Yeah, 0.25 millimolar, 0.5 millimolar, so we'll probably be able to detect between 0 and 0.25 millimolar calcium concentration. Most of us, as I say, most of our sensors are designed to work in the clinical range. What are the recommended current ranges to operate wire sense, hydroperoxide and nitric oxide? Again, I know the wire sensors and the screen-printed electrodes have different geometries, but there should be of a similar order of magnitude in terms of currents. So if I look at the nitric oxide, you can have a look at the signal there. So here's a microamp. Lots of biosensors end up being in the sort of nanoamp, hundreds of nanoamps or microamp range. So here we are, we've got some sort of microamp range here. Let's have a look at the hydrogen peroxide. I think it's because we're actually at micromolars, this is in the nanoamp range. So what do I say? I think most of your signals are going to end up in the nanoamp and microamp range. You have to kind of build for that. And I will go back to here. Do sensors need to be calibrated for each type of solution? Yeah, strictly speaking, they do. Now, this particular person's going to be actually going to be working in cell culture media. I've got a suspicion. So I think cell culture media is fairly. It's not amazingly strange. Like, I mean, we deal with, sources like this. This is kind of highly complex, very viscous. So I got a suspicion. So there's an extreme here, but I think you can do some calibration in just phosphate buffer solutions, and calibrate that way. The ideal calibration is when you actually calibrate in the media that you're intending to use. So if your media is some sort of cell culture or something, you would make up the cell culture and you'd make up some calibration solutions in that cell culture. That means that any kind of matrix effects are kind of caught up in the calibration. I got a suspicion that the solutions that you're using are sort of 90% water anyway. So I suspect that it doesn't really matter too much. It's probably the pH that's most critical, that you should really be calibrating at the pH that you're otherwise intending to be using. So I suspect that it's more important to get the pH right. Most of our calibration solutions, by the way, will probably pH 7.2 because we're all the time thinking about human health. So I'm not quite sure what your pHs are at. So I don't think it's critical. I think it's critical to get the pH right, would be my comment on that. What type of liquid solution is best for calibrating sensors? Well, most of the time we do it in phosphate buffer 7.2. As long as that pH of 7.2 actually is the same as the pH that you're otherwise expecting. Yeah, most of the calibration curves that we have online are actually for for screen-printed electrodes rather than wire sensors. If you're particularly on the wire sensor page, I might have some data there, but most of the data I've got on the website is from screen-printed electrodes. Right, question number 2. Somebody's constructing an analyzer assay. They're buying in some screen-printed electrodes, not from us, from elsewhere, that are pre-activated with streptavidin, and they're characterizing them with ferroferricyanides before use. And they obtain variable results even before they start their process. I think they've got variable results. This is also important to question number 3. This is the big problem in the world of screen printed the world of screen printed electrodes. It's actually variability. So there's a lot of manufacturers out there who manufacture, and then the screen printed electrode they make is variable. So when you get then functionalize a variable electrode, you just end up with a variable product. So biggest problem in screen printed electrodes is actually variability, which is probably, you could actually say, is quality. This is also going to impact question number 3 as well. So, but with question number 2, you're trying to make this antibody sensor. If you come to one of our demos or webinars, you'll often see that we do webinars and demos, and we go through the whole kind of process. But what you'll see is there's no calibration, and the reason that we get away with no calibration is because actually we do a lot of testing. So before we ship a screen printed electrode, we've already tested it in a mixture of sort of ferrocyanide. So the kind of quality control that people are normally doing, actually we've already done it before actually it's even left our facility. So we, for example, we wouldn't ship this sheet because we didn't get enough reproducibility, but we would ship this sheet, we would ship this sheet, we would ship this sheet. And we check the relative standard deviation. So that's the repeatability. People use that word repeatability. I'm not sure that I might want to use the word reproducibility, but we check that before we actually ship it. So, this is single digits. Relative standard deviation is 6.566%. It's really important. So we don't often do calibration necessarily when we're shipping our true products, not the R&D products, not the stuff that we put on the website, but the screen printed electrodes, for example, are these are definitely high volume products from ZP, and the screen printed electrodes, we definitely do a lot of in-house testing. Things like the biosensors, which are a much more R&D product. Now, admittedly, we don't have repeatability on that. But the question here, number 2, is actually about screen printed electrodes and how to make biosensors from them. You've found out yourself that actually underlying those screen printed electrodes that have the streptavidin coating are not repeatable. That is not a shock to me. That's been, that is a big problem, and it's really holding back R&D. We do have a webinar specifically and demo for academia, which you'll see in one of the earlier slides. Definitely come to that. So I'm actually suggesting that, and when you see our results, you'll see that when we do concentration versus signal, for example, we get really good reproducibility, really good relative standard deviations, because of actually we're really good at the repeatability part. I actually think you can't rely on those electrodes. You cannot fix the problem that you have. The problem starts with, I think, an irreproducible electrode, and then you're trying to build coatings on top of those irreproducible electrodes. I think keep it simple. You'll have to build the sensor up from scratch. This is a paper. Here's the link here. I think the paper is free. And follow this paper. Here you'll find that we use a carbon electrode. I will obviously recommend one of our screen printed electrodes. You use pyrene NHS. This is a really easy coupling. We put the antibody on there, and then we block with chitosan to stop non-specific binding. So the first thing that we did is, we make sure that the electrodes are repeatable, but we already do that in our factory. And then as we do each layer, we then follow the process by cyclovoctametry to make sure that we are actually coating the sensors. And we are also checking, in this paper, I think we were actually using ferrocyanide, and we were using differential pulse voltammetry to detect the signal. So your problem, I think, is almost fundamental, that the screen printed electrodes are not necessarily as reproducible as you would expect, or repeatable as you'd expect. Therefore, that coating that's gone on them is going onto a non-repeated sensor, and therefore is then entering into your research. You can't fix that problem. That is something that's going on in the factory now at Zimmer Peacock, because we're much more commercially focused. We really need these screen printed electrodes to be repeatable, not for you, for us. We're the biggest users of our screen printed electrodes. It makes us completely different from everyone else on the market. Who uses ZP screen printed electrodes the most? It's ZP. And that's what makes a difference. Where everyone else has got this kind of business model where they make screen printed electrodes and you're the first person to really test them. Whereas at ZP, we've actually done all that before it ever even reaches you. So I'm going to make some recommendations, A, on the paper, and then B, on the equipment, because I know what you need a connector for a screen printed electrode from us. This connector it takes what we call 7mm screen printed electrodes, and it will also take 2mm banana plugs. And I know that you've got banana plugs at 2mm, and these are the screen printed electrodes, and I'll put links to those as well. Question number 3. So question number 3, they're making an in vitro diagnostic cartridge, and they're happy with the screen printed electrodes, but they're just telling me, look, I get these gaps, and these gaps will lead to, I call it wicking, that solutions love gaps, and solutions will just flow away from the sensor. So they're asking us, and yet they are using the 501 screen printed electrode. Great. I would say, don't change the screen printed electrodes. Actually change that well. Now, I would say that the webinar I do for academia, so if you look at one of the first few slides or look underneath this video, I'll put a link to that, really gives you a thorough explanation as to the problem with screen printed electrodes and the problem with asking for bespoke screen printed electrodes. But I would say, don't change the screen printed electrodes, change the well, and that would be my first recommendation. I will have to back up why I say that, but we do get a lot of requests for alternative designs in screen printed electrodes, and the problem that I see is, this is a design that we got. The problem with these kind of things is, is people want it for low cost. So that screen printed electrodes, people think, yeah, they have low cost. That means that there's plenty of vendors on the market that will give you low cost screen printed electrodes. But the problem with bespoke electrodes and the problem with other screen printed electrodes from other companies is that most of the time they're actually producing them in low volume. So even though a screen printed electrode could, look fairly beautiful, now obviously this is one of mine, it is fairly beautiful and it will be high quality. The problem is that they can look okay, sometimes they actually look terrible, but you're trying to pay low cost. They're not really manufacturing high volume. So when you've got low cost and low volume, you end up with low quality. So the biggest problem that's going on in screen printed electrodes today is actually low quality. In question number 2, they're saying, I'm not getting reproducibility even before I start functionalization. They're actually reporting a quality issue. Why is this quality issue coming about? Because the electrodes were fairly low cost, and actually they're actually fairly low volume. That it's only Zimmer and Peacock where we actually sort of try to discourage people from doing customization, because once you start doing customization, you get something that you asked for, but they're not telling you actually that you didn't pay much for it, and it's producing very low volume. So there's not a high quality. At Zimmer and Peacock, we're able to get high quality in our screen printed electrodes because we keep producing them in high volume. So people say, oh, I want customization. And we're the only company that says, really, do you want customization? Because do you know it's going to be low volume and low quality? And we're the only people who tell people straight. It can lead to low yield. So I was dealing with a company where they went for the lowest cost, and then when they tested the electrodes as in incoming quality, they found that only 33% of them were usable by them. So you'll end up paying for that quality and yield really through effort. So let's say question number 2 is actually buying materials, they're testing materials, and then they're finding they're not repeatable. They're paying for those electrodes actually through their effort. Now, when you're doing your PhD, nobody really cares about your time. It's really bad. What I mean by that is, but you care about your time, but actually because essentially you're not making so much money, people don't really think about the quality. But of course, when you're in an industry like us, any time we're wasting because the materials are low quality is really costing us time in terms of salaries. So you are paying for the low quality, often in screen printed electrodes, but you pay for it through PhD and postdoc time. So it's sort of a little bit hidden from people what's actually going on here. Sometimes people have tried to do designs that are too big and too complex. Now, with the design that we've been asked for, I would say that it's possibly too big. just out of interest, the one that I'm using in this image here, that ended up, that company ended up going into insolvency. And I think they really had a problem of actually, they were killed by the screen printed electrode they were trying to manufacture. They wanted it low cost, so they got it low cost. It was produced in low volume, not by us, but by their vendor. So they got it in, so it was in low volume. It ended up being low quality. It ended up having a yield of 33%. They ended up putting a lot of effort into then testing. It was too big and too complex, and it leads to, in the end, failure. Now, this is the design that the guys want us to do. They want us to basically increase the size. This is our normal size. So the area of the, let's say, new electrode they like us to make is 334 millimeters, and the area of our current electrode is 189 millimeters squared, I should say millimeters squared. That ratio is 1.76. So the one thing about point of care in vitro diagnostics, the mantra is, oh, and I want it low cost. So what you're doing here is by increasing the screen printed electrode part, you're intrinsically at least raising the cost of this by a multiple of 1.76. Now, because you'll be then a bespoke electrode, you're going to end up having to pay more for it. You'll end up being in low volumes, and it will actually lead to low quality. So ZP, we will do you custom electrodes, but we will warn you, and you will find, I think there's over 50 houses out there doing customization. But what nobody's telling you is, because you'll want to not pay lots of money for it, so you will pay low. It'll be low cost. It'll be very low volume. You may not even want more than 3,000 of these things, which is no volume, right? Volume is really 100,000. So you'll be low volume, and therefore low cost, low volume, low quality, and intrinsically, because you're trying to go bigger, you're trying to fix, let's say, this engineering problem by making the screen printed electrode bigger. But you're intrinsically then making the cost you're increasing the price by 1.76. This is because we're producing them in sheets, and so you're taking up much more area, and so our density of manufacturing just went down. If anything, actually, with your design, I would actually be making, if I was really commercializing this, I would actually be making it smaller, so you've got more screen printed electrodes per sheet. But the problem is the kind of run, a manufacturing run from Zimmer and Peacock is 100 sheets plus. So you could end up with tens of thousands of pieces that you will never use. So I would actually say change the mechanicals, don't change the screen printed electrode. So question number one, wire sensors. Sorry that I only just really gave you verbal answers today. Like I said, I'm going to have to catch this flight in a few hours. I'm also going to have to pack the bags. I'm a family guy, so I get balance family on the weekend and also, unfortunately, a business trip. So we've got wire sensors to deal with, which we have dealt with. Immunosensors, you cannot fix that problem that you have. You've just found one of the fundamental problems of screen printed electrodes and functional screen printed electrodes, that the repeatability or reproducibility is really not great. That webinar, especially the webinar I do for academia, is a really good one for you to come by. I've recommended an electrode, I've recommended a connector. You have to get them. Making a custom screen printed electrode, I think a lot of commercial development and a lot of academia has actually been held up by this customization problem, because it leads to low quality, which means that you end up doing lots of testing and a lot of throwing away of data and a lot of heartache, because when you've got poor reproducibility of screen printed electrodes, when you're then trying to build a biosensor, you're never sure whether you're making improvements, or you can't see an improvement. You've changed a formulation. You say, is that formulation better than my previous formulation? But you don't know, because the noise in your data from the irreproducibility of that underlying screen printed electrode is hiding your improvements. So be really careful about customization, especially customization too early on. Any links that popped up today, I'll make sure I put them underneath this. If you're just watching this webinar and you've got some questions, don't hesitate to reach out to me. As I say, we do like to go live, but I don't know, my bandwidth was looking really bad this morning, so my apologies for that. And I have to go and catch a flight as well. So take care and have a good day. All right, thank you so much.